one went abroad in India. And so now it will be to talk about the Himalayas.
Thank you very much, Zdenka, for introducing me, for inviting me. It's really a great honor. I've been here 12 years ago, I think, in 2010, to give a lecture about the research in Himalayas, but it was really introductory because we were just 2 years working there. So now a little bit more about what we are doing in Himalayas, about the Alpine plants. And this is the overview of the things I'm going to talk today. So I will give you, at the beginning, some sort of intro into a broader context of Himalaya, because if we say Himalaya, most of us can imagine snowy peaks, glaciated, lush forest underneath, maybe with the Yeti. Everything is basically because the Himalayas are stretching from northwest to southeast, and they are blocking the summer monsoon that comes from the south, and they are unable to cross the mountains. So the territories north of the Great Range are kind of dry, ranging from desert to steppe to alpine. And it really contrasts with what we see on the southern flanks. Just a little intro into what you could potentially see on the southern slope, because this is really amazing, a sequence of different biomes that are compressed over short distances. So, and the diversity is not necessarily following the linear decrease with elevation. There is some sort of mid-elevation species richness peak. At the very low elevation, we can see the tropical forest, not the rainforest, the semi-deciduous forest, because there is a protracted dry season lasting from 5 to 8 months. And this tropical deciduous forest is followed by a very species-rich subtropical or temperate oak forests that are one of the most diverse ecosystem on the southern flanks of Himalaya, and also those that are most threatened by human activities, agriculture, farming, pasturing. We cannot see them anymore, or in rudiments in Nepal, for instance. If you want to see them, go to Bhutan. If we climb higher up, we may get to the different types of conifer forests depending on the precipitation. On the wetter side, we can encounter Himalayan hemlock forest, Tsugadumosa, which is one of the largest living tree species in Himalayas, with the known specimen being almost 800 years old. On the drier places, we can see the cedrus forest admixed with the pine forest, Pinus vallichiana. Higher up, where we have more precipitation, there are spruce forests of Pizza smithiana, pretty much as our spruce forest. And even higher up, we can see the fir forest with the Abies spindro or Abies spectabilis. And above this conifer zone, we can see the Betula utilis forest, the birch wood that is admixed with the shrubs, with the rhododendrons. And what is important is to say that the alpine zone that follows above the shrub and birch tree line is relatively narrow. Because it is delimited by the forest at the lower elevation and by glaciated peaks in the higher elevation. So compared to what we will see in the northern part of the Great Himalaya on the southern flanks, this alpine zone is relatively narrow. And you may find very peculiar alpine plants that evolved to cope with both the dry condition that prevail in spring before the monsoon arrives, and at the same time that must somehow cope with a lot of snow and rain that comes during the summer monsoon. So they develop sort of protection against the surface wetting during the monsoon plus radiation shield during the dry season, often using the bracts. So here you can see the representative of Asteraces, Sausurea, and Marmoritis to the right. Another species that you can encounter is this Saussura medusa. Again, you can see these woolly leaves that protect them against the high UV radiation and also against the recurrent snowfall during the summer season, or this Saussura bracteata. So these southern flanks of Himalaya are fed by the summer monsoons. This is not true about the mountain ranges north of the Great Himalayan Range, like Tibet or Karakoram, that are deprived of summer monsoon, and they rely on the winter precipitation, so-called winter westerlies that come from the north west. So when we cross the mountains and get to Tibet, part of it is the region called Ladakh. It's part of the so-called Trans-Himalaya, and it's sandwiched between the Great Himalaya to the south, Karakoram to the north, and Tibet to the east. So it's pretty diverse in terms of geology. And it's dry, as I said, because the monsoonal clouds are blocked by those 6,000, 7,000, 8,000ers. So this is the picture from above. You see glaciers, and suddenly we are on the northern side. The peaks are less and less glaciated. And Ladakh itself is dissected by the Indus River, which is a backbone of the country. And it is basically the delimitation of the Indian and Asian plates. So they collided where the Indus is now. This region is historically very interesting, rich. It's one of the few places where we can still see the traditional Buddhist landmark sites, plant life, the human life, etc. And it used to be bigger. It encompassed in the history also what is nowadays the part of the Pakistan, the Baltistan, where we have the 4,000ers, 4 8,000ers, including the K2 and Broad Peak, etc. And part of the Ladakh used to be the Aksai Chin, which is now a part of the China. As I said, the whole region is geologically very diverse. We have everything from sedimentary rock to granite, batholiths, etc. And this also makes the plant diversity relatively high, given the overall dryness and high elevation. So the Indus River, as I said, is the backbone of Ladakh. Here you can see an example of the sedimentary rock, different conglomerates, dolomites, like this. And similarly, as on the southern flanks of Himalayas, on the northern side, we see that plant species diversity is not decreasing with elevation linearly. It has some peak at lower elevation. And I will get to this a bit later. Just this entire flora of Ladakh have different origins, so we can basically divide the native species into the Himalayan elements, which forms, for instance, 20%. Then we have 15% of species that are coming from the Central Asian steppe zone. We have some endemic species, some Tibetan elements, some species that have roots in Pamir, etc. So the entire flora of the region is composed of plants of different floristic origin. And compared to southern flanks of Himalaya, where we have everything from tropical forest to alpine zone, in Ladakh or in northern territories, it's different. At the lowest elevation, we have basically desert or semi-desert. Like here in the Nubra Valley, confluence of two mighty rivers, Shyok and Nubra. This is also the place where the villages can be found with the arable land and fields and etc. And following the desert, the most widespread type of vegetation are those alpine steppes. And higher up, we have the alpine meadows or alpine vegetation as we know it, for instance, from the Alps. And higher up, we have a glacier, the upper alpine zone, subnival zone, which is the fertile land for grazers, for yaks. And this entire region was inhabited for quite a long. We found these rock carvings, so-called petroglyphs, at high elevation, 5,200 meters, which indicates that probably Neolithic people were coming to these places several thousand years ago to hunt the game, and they depicted the different animals and hunters on the rocks like this. It also indicates that this valley wasn't glaciated for long, probably. And subnival zone, an example of it, with the snow patches and sparse vegetation. So Ladakh is also the place where we can find the highest occurring angiosperm on the planet. So we found the plants at above 6,000 meters, at 6,150 meters. One of them was this Sausurea gnaphalodes. So the Ladakh is the place where we have the broadest alpine zone, or let's say the treeless zone, if we also consider this alpine steppe as a part of the let's say, treeless alpine in broad sense vegetation. So this was for the introduction. Now let's dive into our research and results. So we were interested in exploring very basic ecological question related to species richness and species distribution across elevation gradient. And we tested if species richness has a peak along this vast 3-kilometer long elevation gradient, how individual species are distributed across the elevation gradient, and if the range sizes show some non-random pattern. So for that, we used the data that were collected largely by Dr. Klimesh, who started his research already in 1987 and was visiting this territory every season since 1997. And unfortunately, he died in Ladakh in 2007. So he left a tremendous amount of data on species distribution. So we added something, and then we tested the diversity distribution. So we were interested if they follow sort of neutral, random pattern, or if there is some non-randomness. So we compared the distribution of species with the prediction generated from the mid-domain effect theory, and we didn't find much support for it. For us, important is that the peak species richness is shifted towards the desert, which is kind of surprising because we would not expect species richness to be highest within the desert zone. But it is true, and it is largely related to the fact that unlike other drylands, other desert, the Himalayan drylands are intersected by rivers like Indus, like Zanskar, like Nubra, like Shayok, and their tributaries. And these rivers are more or less permanent. They are not drying out like other rivers in deserts, so they can sustain plant life more than we would expect just based on amount of precipitation, which is 50 millimeter to 100 millimeters annually. So in other words, within the desert belt, we have a huge diversity of habitats, often bound to riverbeds, the riverine vegetation, that increase the total diversity. This is an example of the species that has optima at 4,500 meters, and then towards the higher elevation quickly disappear, but it has a long tail towards the lower elevation, which means that this species is pretty much descending along the river down to the valley, and bring the higher diversity within the desert belt. So we were also interested how many of the species like this can we find in Ladakh, because we could expect that species has a normal symmetrical distribution, but how many species has asymmetric niches, like this one? So we tested that, and we were also interested if we can find a certain elevation where species with asymmetric niches may concentrate. If there are some aggregation of, for instance, upper elevation limit over short distances, that would indicate a transition from one biome to the others. So we fitted almost 400 species with enough occurrences in our database with logistic regression, categorize them based on the response to the flat, left-skewed, symmetric, right-skewed types. And we expected the aggregation of species with asymmetric niches both towards the drier, lower elevation and towards the colder upper elevation, and species with a symmetric distribution to have someone in between. So what was the result? We found actually that, and we compared the distribution with the null model, with the generated the distribution of different types. And we found out that 36% of tested species have asymmetric left skew distribution, and they are actually concentrated in elevation around 5,000 to 5,400 meters. The species with the symmetric responses were found more likely at the lower elevation, and species with the right skew distribution made up only 2.3%. What is most interesting is this. So what is this elevation about? What kind of species can we find here? This is the elevation where steppes are turning into the alpine vegetation. So this is how those steppe may look like. Another example of rather dry, semi-desert steppes with the pebbles, with the stipa, with the oxytropis, with the artemisia species. Dry steppe with the grasses on the sandy deposits, and they are at around 5,000 to 5,400 meter transitioning into truly alpine vegetation with the sedges, with the cobresias, with the grasses, and with many forbs. Sometimes they form really diverse communities. We can find plenty of species coexisting. Last year, we did new plots, and in vegetation like this kind of upper alpine, almost subnival vegetation, we can find up to 35 to 40 species per square meter. They're really tiny, tiny, tiny. So, and of course, alpine cushion plants in the highest positions. We were also interested if the transition from optima to minima is getting shorter with increasing elevation, and we found some evidence for that, which means that with increasing elevation, with increasing risk of being exposed to freezing temperature, there is a sharp transition from optima to minima. So those response intervals are getting shorter, and again, we can see that in these elevations, there is really something going on. It's that zone of when the plants that are bigger, those bigger subshrubs, shrubs, artemisias are turning into low stature forbs and grasses. So what is the take-home message from this study is that plants cannot escape cold, especially in these plains where we do not have big rocks, we do not have the chasmophytes. They cannot protect themselves. So they are getting smaller and smaller. So they cannot escape the cold, while plants at the lower elevation towards the dry steppes and semi-deserts then can escape the water stress because there are the rivers, there are riverbeds. That's why we do not see accumulation of species with asymmetric niches at the lower elevation. Rappaport's rule. You have heard about Rappaport latitudinal or elevational rule. So this is something that people were testing for really long, and it is generally related that with increasing elevation or increasing latitude, we have much higher temperature fluctuations. And so the plants should be basically adapted to the wider range of condition, and therefore they should have a wider elevation or latitudinal ranges. Can we find something like that in our condition across the 3-kilometer long elevational gradient? So the Rappaport rule was confirmed in many cases, and it's usually associated with a decrease in species richness. We found a support for the Rappaport elevation rule on Mount Cameroon for trees. Just a little side track. You can see that elevation ranges estimated by the Stevenson method are basically increasing with increasing elevation, while species richness is linearly decreasing. But this is a tropical island. Mount Cameroon starts from the coast and goes to 4,000 meters. So it's pretty much delimited at both sides. But how it is in Himalayas, because the Ladakh doesn't start at the sea level, we have the lower elevation at around 3,000 meters, 2,800 meters. So the ranges of many species are in fact truncated. You can see it on this figure, where we have species ranges both within the Ladakh, this dark blue and light blue, including the subcontinental ranges, like full ranges. So this comes from our data, and this we took from the literature. And just from this picture, we can see that there is not much going on. There is no really... They are smart, those smartphones. They didn't get the name by chance. Sorry. So I don't want to spend much time with this, but we didn't find really support for Rappaport elevation rule. So the species ranges are not really going wider with increasing elevation, both for the global and regional species ranges. So no support for Rappaport's rule. Just an illustration that even at the highest elevation, where we would expect species to have narrow ranges, to be really a specialist, we can find among the best climber, those that goes over 6,000 meters, those that have both very wide ranges, like Waldheimia tredactylites, which can be found 3,000 meters lower in the riverbeds. This is this species. You can see that it has a range that goes from 3 to 6. The same for Po attenuata, but there are also species that have narrow ranges, like Ladakia lacrymeshi, the species that was discovered by Leoš, or Sausurag naphalodes. So those local communities are composed of species that have both narrow and wide elevational ranges. And one of the explanation for the Rappaport's elevation rule, as I mentioned at the beginning, was that the temperatures are fluctuating with increasing elevation more than at the lower elevation. We didn't find anything like that. We look at the seasonal T range or diurnal temperature range, and in both cases, there is no really significant pattern that would probably or could potentially explain the things. So to sum up, mid-domain effect versus environment, that we didn't find really much support for mid-domain effect. Rappaport's rule neither. Asymmetric abiotic stress limitation hypothesis, asymmetric niches, yes, we did find a support. So now let's move to other topic, which is what can determine the species elevation optima and limits. Yeah, we talk about distribution of species, about the species richness distribution, but what is behind the species optima and species limit? Based on those asymmetric niches, we found out that there is some aggregation of upper limits, and it's probably related to the low temperature stress. But what it affects, because low temperature can affect a bit of everything. It can harm regeneration, it can harm growth, it can affect survival, and even for the growth, it can affect thousands of things. So we were interested in this and did a case study asking what determined the tree line in Ladakh. It sounds weird because there are almost no trees in Ladakh. So why we should call them tree lines? But by definition, tree is something that is higher than 3 meters. So this big shrub or small trees, because it can be both multi-stem or single stem, can go up to 4,400 meters. And we were interested if the upper elevation limit, the formation of alpine tree line, is related to low temperature stress on growth, or for instance, carbon assimilation, because the reason why the plants cannot grow higher can be related to the fact that it doesn't have enough resources, including assimilates, nutrients, or they can have everything in plenty, but enzymatic processes are limited by low temperature and growth is not performed. So we can test this by looking at the growth variation across the elevation gradient and also the variation in some proxies of resource limitation, right? So first, we were interested where exactly we can find the upper elevation limit of this guy. Is it really at the places where normally tree line occurs, which is something around 6.7 degrees of Celsius of mean growing season temperature? All the tree lines, those natural tree lines that are not depressed for whatever reason, fires, volcanic activity, usually occurs where mean growing season temperature is around 6.5 to 7 degrees of Celsius. And this is set to limit primarily growth. Okay, so we put the data loggers and we found out that indeed the upper elevation limit of Myricaria is at this temperature. Growth was decreasing irrespective of the plant age or plant size. So the growth at the tree line was really low. And what was mainly affected was the early wood. But when we look at proxies of carbon limitation, nutrient limitation, by measuring concentration of nutrient in leaves, concentration of sugars in different organs, branches, leaves, stem subwood, we didn't find any support that tree line trees would be limited. So they are fully charged, they have enough water, they have enough nutrients, they have enough carbon, but they cannot invest it because the temperatures during the growing season are low. It's something like you would like to construct the house. Yes, you have money. You have all the building material. You have even the bricklayers, but it's cold. And the bricklayers go on strike. Instead of starting constructing your house, they sit in the cabin, drink the rum, and do not work. So this is, the bricklayers are the enzymes. So they have higher temperature threshold to be active. Photosynthesis can, as we know all, pretty much work at really low temperature, even slightly above the zero, but enzymatic processes or meristematic processes, cell division, cell maturation, cell lignification, is something that needs higher temperature. For lignification, for instance, the threshold temperature is between 6 and 7 degrees of Celsius. So that's why we do not have the trees when the meat growing season temperature is lower. Anyway, what is most affected, because the growth is really a black box. So, but if you look at least on two parameters that are distinct, early wood and late wood, in this case, in case of Medicaria, because it's angiosperm, it has very distinct early wood formed by big vessels that are produced during the short period of time, followed by heavily lignified late wood. But there is a problem very often. Almost every other year, there is this happening. This is the result of late spring frost that affect the plants. Then the whole year increment is kind of narrow, and this contributes in a way also to a tree line formation. So the early wood is most sensitive because in this dry subtropical region, the spring starts very quickly. You come there in late April, and within 10 days, everything has full foliage. But temperature can drop below zero till mid-June. So those plants are exposed to night frost. So that's the result, and the result is the wood deformation. This may eventually lead to cell cavitation, embolism, and leaf wilting, but usually Medicaria is tough enough to survive those things. Another story, what determines species elevation optima? So we were interested in finding some easily measurable traits that could, in theory, explain why some species has optima here and the other there. We know that plants are primarily driven by temperature. And we ecologists often use the temperature to predict what will happen with the species. Yes? So we expect that there is a direct link between plant performance and temperature. There is 1,000 of papers that are showing us that if the warming will continue, the species will move upward, polarward, etc. But is that really true? Are the plants that silly to directly respond to temperature? Maybe not, because they evolved in a way to reduce the extremes. So they try to be decoupled as much as possible. And this is particularly true for the alpine plants. They don't want to live in the cold environment. That's why they are small. They try to hide themselves. And they can be substantially decoupled from the ambient condition to perform carbon assimilation. But how to measure the leaf or plant temperature to see how much they are decoupled from the ambient condition? It is technically very difficult, yes, especially if you would like to measure tens of species over the entire growing season. But there is a tool or approach to bypass those difficulties, and one of them is that we can use the stable oxygen isotope theory. We can measure oxygen concentration in cellulose, and then using a modeling, a lot of other variables, we can actually calculate the average plant temperature or leaf temperature during the photosynthesis. We read those papers that were published by Chisong and Heliker, and they found out that plants are able to thermoregulate. They demonstrated it across the latitudinal gradient from tropics to the boreal zone, and they simply discovered that trees that are growing in tropics must cool down to reach the temperature around 20 degrees of Celsius, and boreal tree species have to warm up. Can we see something similar in the Himalayan plants? Are they really cooling down by evapotranspirative cooling at the lower elevation within the desert? And are they really warming up substantially in the higher elevation? So we collected about 70 species. We measured the oxygen isotope and estimated the leaf temperature. And we also measured at the same site the ambient temperature using the data loggers. And so we could calculate the deviation, so the extent of decoupling. So what's the difference between the plant temperature and the ambient temperature, okay? And then we said, okay, but this level of decoupling can be related to the leaf thickness, because this is a proxy of the heat storage and transfer. Yeah, the thicker leaves can store more heat, and vice versa. Or the level of decoupling can be related to the how plants cool down by evapotranspirative cooling. So we can use some proxy. We use the water use efficiency approximated by the carbon-13 isotopes. And again, the plants that have enough water to cool down, they have lower water use efficiency, plenty of water. While those plants that are coping with the water shortage, they close the stomata, and that's why they have higher concentration of the isotopes. And the height, of course, the plant size. This is exactly related to the plant decoupling. The big trees are pretty much coupled with the ambient condition, while the small one not. So we use those three traits to predict the TL and delta T. And then we thought, okay, but could those things explain the species distribution, like optima? Because we have this information for every species. So we did that. What was the result? So we indeed found out that plants have to warm up in dry condition and warm condition, have to cool down, and vice versa in very cold condition. At the Alpine subnival zone, they warm up. Yeah. So, and then we measure the traits, LDMC, plant size, and water use efficiency, collecting the plants, sorting them, etc. in the field. And then we made a correlation or regression, and we found out that indeed leaf temperature is pretty much as predicted related to leaf thickness, LDMC. The level of the extent of decoupling is pretty much related to the plant size, and the same applies to water use efficiency as predicted. So then we made a regression between TL and delta T and species elevation optima, minima, maxima. And we found a good match. And the last step, we took another 100 species for which we didn't have the oxygen isotope to derive the TL and delta T values. And we basically use it to validate. So we calculated the predicted elevational optima, and we compare them with the observed. And we found a reasonable relationship. So to conclude this whole story, so there are relatively simple traits that can predict the extent of decoupling of leaf temperature from the ambient, and this is pretty much related to the species distribution. So, plant adaptive strategies. So this was partly about plant adaptive strategies, but there are plenty of others. Of course, the most easily, or let's say measurable, are the different growth form. So let's now jump a little bit into those, because they are very important and they explain a lot of things. So I was showing this when I was here 12 years ago, but let's rehearse a little bit. So this entire plant diversity, or can be categorized into about 20 growth form. And Ladakh is interesting in that about 60% of the species are non-clonal. So they retain main taproot. And this is important to say because this allows us to, for instance, look at the absolute age of the plants, because those plants are perennial, they have a main taproot, so we can cut them, we can make a tiny cross-section and count the rings. So clonal species are rather underrepresented in general because of overall dryness. And they form about 20%. And we have a lot of different growth forms depending on the type of rhizome and below ground systems. So I'm not going into the details, just for you to know that about 60% non-clonal, taprooted, 20% clonal, and only 15 to 20% are short-lived species. That's why Professor Mihe is not calling this environment cold desert, because desert by definition should have at least 50% of annual species. And he calls it Alpine steppes because of a predominance of perennial species. And those different growth forms has, of course, distinct ecological functions. So those cushiony plants, they often ameliorate environment. The plants with the longer rhizomes, they are found in the wetlands, and they are very efficient in space occupancy. Plants with the deep taproots, they often occur in the dry places. Those taproots can provide access to water. What is very important plant growth form or strategy is this one. We call it pleocorm. So there is a lot of plants that have small deep roots and long below ground branches, and those plants are very efficient or successful in unstable substrate, in the screes, talus, etc. And root sprouters, the plants that reproduce by adventitious roots, they are very often found in sandy and gravelly deposits. And a few examples. So you can see that plant has rosettes. They have a very peculiar anatomy. Most of the high alpine plants are composed of cells that are living, parenchymatic. Many of them are quite old, and they still retain living cells. Why they do that? There are at least two reasons to mention. One of them, why they are largely composed of living, unlignified cells, and that's why everything is stained blue, yes, is that they can store carbohydrates in living cells, so they can protect themselves against cold and drought. Another reason why they may use the living cell is that if something happens, some disturbance will come, they can activate those living, very basic parenchymatic cells to seal the wound. So something that you would consider to be very short-lived, like this species, when you dig it out, you will find out that this guy can sit here for years or decades. And you say, should I dig it out? It's like, chopping down the large trees in the old-growth forest. So we should be really careful about that. And they have a beautiful anatomy. Look at this. So this has a kind of caudex. Let's call it caudex. It's woody. And that's why we see the xylem largely lignified. But still, there are huge rays. So this plant has a capacity to store, and at the same time, it's pretty much lignified. So another example of species that is rather bound to the wetter places, the really northern flanks of the Himalayas with more precipitation, those large bracts protecting the flowers by maintaining dry environment and luring the pollinators. Tiny primula, Tilaco. There are species that can live in the water stream, but those situations are not that prevailing in these dry lands. Scutellaria heydei, you would say, this is a short-lived taxa. But if you look at the anatomy, you think, if I would see this without this, I would say, this is the oak. Yeah, so you can see the same structure as in oak. So this is a ring porose species. You can see the huge early wood vessels followed by the small late wood cells with heavily lignified secondary cell walls. And of course, the plants that grow within the screes, they do show this kind of eccentric radial growth, similarly as our maple or beech trees on the steep slopes in Carpathians, in Tatras. Some of the plants, like this endemic sagebrush, Artemisia, may have really huge capacity to store the carbohydrates. As you can see on this multiseriate rays on the tangential section, maybe this is the reason why this plant is one of the oldest living in Ladakh. It is partly clonal, but those living fragments can be up to 80 years old. Another very conspicuous species is Delphinium brunonianum, which is a tall forb bound to protected screes growing among the big boulders. And this enables him to go up to really extreme elevation, 5,700 meters, but always found where we have some big boulders, always in places which are providing some sort of better microclimate. But this species, and also this one, they are interesting by doing something that maybe is better illustrated on this picture. You can see that they are not wasting the biomass. You know, growing in these conditions means that resources are limited, but if they grow like this and become bigger, this was first a small plants. As it grows bigger, those individual tillers compete with each other. So there is a tendency for the main taproot to segregate. But they don't want to waste them, so they get splitted. But these plants come with a trick how to protect themselves, because the splitting the roots means that this inner part become naked, and those plants can be attacked by the fungi or bacteria. So the plants want to protect themselves, and they activate those living parenchymatic cells to produce a new periderm. We call it intraxillary periderm. So at the end, the entire perimeter is protected by the bark. So what was basically before in the very pith, in the center of the plant, and become separated, gets a new cover. Yeah? And this formation of intraxillary periderm is very common in unrelated species. So this is the ranunculaceae, this is asteraceae. So you can see here. Another example, again, the nice combination of lignified and unlignified tissue, because these plants want to store at the same time, must get lignified to be able to keep those big above ground shoots in upright position. Pleurospemum candolae, it looks like a tree, huh? Because the picture was taken from the ground. But even this umbelliferae species can be up to 17, maybe 20 years old. Another example of the semmering porous wood structure. So to speed up, anatomy. So anatomy is a way to understand the behavior of the plants, adaptive strategies. We can then play with the data, as we did here. We can do or depict some trends. For instance, with increasing elevation, we have increasing proportion of species that have semmering porous structure, that has little lignification or lignified tissue, and they have a really big prostrate race to store the carbohydrates. And then we can test how much this interspecific variation in anatomical settings is related to the growth form, plant size, or environmental forcing. And this is just to illustrate that most of the interspecific differences in anatomical structure can be rather explained by simply growth form. That's why it's important to make this categorization. But there is also a net effect of elevation or moisture gradient. Just let's stay with that. I am not going to go into details. And then if we have this kind of data, for instance, the extent of lignification, we can do some global study. We can see if there are some trends. So here we took 1,800 species and the extent of lignification. And we found out that indeed the extent of lignification is decreasing above the tree line dramatically. So this is just an illustration how we can use the data. And we can do something similar with the grasses or monocots. We may not restrict ourselves in this anatomical research only on dicots. And we can again test something similar for monocots. So we took about 150 grasses and sedges from Ladakh and from different biotopes, like those Cobresia loans. This is, according to some opinion, the species with the largest land cover worldwide. Okay, let's believe the source. But it is also the habitat that is really endangered because of increasing stocking rates in Ladakh. This overgrazing is terrible, and this is an example how it may look like when it starts collapsing as a result of overgrazing. Yeah. So we can basically quantify different anatomical feature on the cross-section. We can even go further to just simply quantify what is the proportion of lignified, non-lignified, and conductive tissue. And then we can look at the trend if they follow what we have seen before for the dicots. Yes. So for instance, for grasses, we can see that the species that are growing at higher elevation have lower proportion of conduits and larger proportion of storage tissue, similarly as in forbs. Again, having smaller vessels is something that may protect plants against freezing-induced embolism and cavitation. And again, for the monocots, we can look what is the proportion of variation in individual anatomical feature explained by elevation, which means temperature gradient, moisture gradient, growth form, and plant size. I'm not going into details. Another line of research we were following was if we can categorize different habitat types and if we may find the differences in species diversities and, for instance, coexistence mechanism based on the trait structure. Yeah, here you can see 8, if I count correctly, vegetation types. Semi-desert, steppe, water bodies, wet grassland, subnival zone, etc., shrublands. Yeah. The most diverse, so we are talking about the alpha diversity, plot level diversity, are alpine grasslands and alpine screes. With some of the outposts, as I mentioned before, when the diversity goes over 30 species per the basic sampling unit. And we can take the traits of the coexisting species and test if they are non-random, if there is some sort of trait syndrome that deviates from if we would assemble community just randomly. And interestingly, we found out that there is usually high convergence in most of the traits above ground. Not surprisingly, because above ground there is cold, there is a strong wind, there is a high radiation, etc. And not really significant convergence for phylogeny, which means that we have communities that are assembled of unrelated species that converged in their traits to be well adapted to prevailing environmental condition. Yes. So this is the one of the line of research I wanted to say. So, and these trait convergences that we see above ground may not necessarily be seen below ground. Yes, so this below ground space can be less hostile than above ground space. And species to coexist, they may have different root system, different rhizome system. They may acquire resources from different soil depth, etc. There This community type differences can be also explored using something that is rather tedious, and that is plant biomass and nutrient allocation. Plant ecologists are often using the easy-to-measure traits, but better idea about mechanism of coexistence or trait variation can be gained from simply measuring the biomass investment into individual organs. Yeah, we did that for 200, almost 60 species, 1,200 individuals. So we were sitting and putting the leaves into one bag and roots into another bag. And then for the same plants, we measured the leaf nutrients in different organs. And then you can see that there are some trait syndrome. For instance, the alpine plants have more leaf nitrogen, more root nitrogen. Of course, the steppe plants are always bigger. They invest more into the roots. The alpine plants more into the leaves, etc. So this is something that we are now trying to get together and publish, this coordination between investment of nutrients into different organs. And also, this system is unique in that we can also test if the biomass and nutrient allocation to different organs vary not only between the basic habitats, like dry steppes, cold alpine, or productive wetlands, but because these habitats stretches across the vast elevational/temperature gradient, we can test if there are some trends within the habitat. Yeah, for instance, we see that steppe plants are always bigger than wetlands and alpine plants, but even within the steppe, we can see that plants are getting bigger with increasing temperature. So this is just to illustrate that many of the things can be tested by looking at the biomass and nutrient distribution. So I will skip that, and we will get to another interesting case study, which was designed to test if carbon limitation can explain the upper elevation limit of herbs. Before, I have shown you an example of a woody species, Myricaria elegans, and we have seen that Myricaria has enough carbon, enough non-structural carbohydrate as a proxy of carbon charging, even at the tree line. And can we see something similar at the herb line? So do plants at the very, very high elevation, at the upper limit, have problems with the carbon gain, with the photosynthesis, with the assimilation? So my student, Zuzana Chlumská, tested that. She was particularly interested in the different NSC compounds, because this is, again, like a black box. The total non-structural carbohydrate can be decomposed into sugars, sugar alcohols, and polymers, like starch, fructan, and raffinose family oligosaccharides. And they have different function: sugar plus sugar alcohol, cell stabilization, starch primarily as a reserve, fructans and RFO, reserve and cell stabilization. So we took 11 species and sampled them across the entire, more or less entire elevation gradient, from low to high elevation, including the upper elevation limit, from steppe to subnivel. Yeah. And we found out that there is no decrease in sugar charging with elevation. So the concentration either increased or remains more or less stable. So there was no any species that would show a decreasing trend. And even the highest growing plants have a lot of NSC, like Ladakia lacrimeshi or Waldheimia tridactylites. And interestingly, unlike woody species or meadow species in our condition, the fructans and soluble sugar prevail, and less so starch. This can be pretty much related to the drought or frost tolerance. And this was also somehow indicated by the diverse trend of different NSC, this acclimatory function. So this high concentration of non-structural carbohydrate in high elevation plants is not necessarily only sort of passive result of low temperature limitation on growth, as often stated for trees, but can be an active adjustment to cope with both the frost and drought. So anyway, no lack of carbon to explain the upper elevation limit. There can be other reason, of course. So the plants have no issue with photosynthesis. So what is behind the upper elevation limit? What is behind the fundamental niche? One of the reasons is that plants have somehow limited dispersal. So they could grow higher, but they cannot get there. So let's test that. Let's take the plants, bring them to higher elevation, and if they are going to survive there, we could say that the problem is with the dispersal or with the, let's say, early juvenile stages. So if the adult plants will make it, this would be an indication that the problem is with the dispersal or regeneration. So we did that experiment by transplanting the plants from the core zone to upper elevation limit of vascular plants, crazy things, to 100 meters above the upper elevation limit. And we designed this transplant experiment to also include some sort of treatment, like because we expected that plants could better survive if they are protected. So we created shelters. And we also expected that plants could eventually survive better if they have more nutrients. So we have control, shelter, nutrient addition, shelter, how it looks like in reality. We built some fences, fancy fences, stone fences, and we put the plants in them. Yeah. It was beautiful weather, everything went fine. And we measured the microclimate to see how those sites differ. And we were following the survival for another 6 years. We started this experiment in 2009, and every year we climbed to there and look what plants survive and so on. The thing is that those sites are really extreme, and in some years the plants were covered by snow, so there was the problem with the missing season, and the snow can come any time of the year. Let me show you a few pictures, because normally in our mountains, well, except this year, we have snow that may last from autumn to spring. In Ladakh, in these dry lands, this is a normal situation at, so this is taken by the, this is taken by the plant camera that was snapshotting the experimental sites every day in the morning. So there is a little snow during the winter. April, May, there was some terrible snowfall event on the 1st May, but the snow is sublimating, disappearing quickly. And this situation, July, perfect weather, but oops, morning snowfall. If you are in the tent and you have 10 centimeters above your head, that's not pleasant, but it can melt quickly. So this is already the afternoon picture. But this brings a lot of moisture for plants. And if the snow is in surplus, it may not be that good for them. It may actually cause a soil disturbance. So what we found? We found that species adapted to soil freezing and thawing survive better. There are 3 champions, Saxifraga cernua, Patenotata, Saxifraga nanilla. They were the best in surviving. Interestingly, Saxifraga cernua is a species that has optima at really lower elevation. And habitat amelioration, those enclosures, increased survival. Fertilization didn't help. The last survival, of course, at the beyond site, but plants were able to survive for 3 and more years. So this is some indication that maybe they are dispersal or recruitment limited. And important take-home message is that those extreme climate events, those episodic climate events, can really be a decisive element. Like it happened in the spring 2013, long-lasting snowpack, most of the plants died. And we found in the literature that winter of 2013 was catastrophic. Around 24,000 livestock perished due to the extreme cold and starvation, also our plants. Bad luck, but nice illustration that this is probably important, of course. Just a few pictures that, in fact, this result partly negate what I said before, that the upper elevation plants are not really carbon starving, that they have enough carbohydrates. You can see that the plants at 6,100 meters and those at the upper elevation limit are really differing. So maybe those plants are dying because they do not have enough carbon. So it can be really a part of the story. And what makes plant really dying is the frost upheaval. Yeah. So basically, during the summer season, the temperature is frequently falling below 0. So the risk of the mist season, a story of the highest occurring vascular plants. So beside this experiment, we found out that there is an outpost 150 meters above the upper elevation limit. Those 6 species we found at this high elevation, basically 150 meters above the upper elevation limit of continuous vegetation. Let's call it like that. But of course, this site was somehow on the southwestern exposure. The plants were growing among the big stones. So they were doing well. And the temperature at this site was actually much lower than surrounding temperature and corresponded with the temperature 150, 200 meters below. So it's not really the absolute elevation that matters, but local environmental condition. We were also looking if those plants are unique. So we did a study looking at the some kind of helpers that may help these plants to survive in these environments. So we look at the arbuscular fungi and bacteria that can bring the nitrogen. So we did the whole metagenomic to find out that those plants do not rely on fungi, but they have a diverse set of bacteria. Some of them are able to fix atmospheric nitrogen, which is probably the most limiting element in this elevation, unlike phosphorus. So this screening we did, or screening for fungi, we did for another species, like 62 species. I'm not going into the details, just to say that, interestingly, plants that are normally having fungi or AMF didn't have them above 5,800 meters. So we found the AMF up to this elevation, but plants could grow higher without them. So reason is probably that maybe they do not need them, because phosphorus is readily available in this elevation, because there is water. At lower elevation, there can be phosphorus, but it's bound because the soil is alkaline and dry. Or another possible explanation is that they don't want them because it brings additional cost to provide them with the carbon. So maybe it could be a combination of both, and experiment would be needed to tease it apart. But those higher elevation plants had endophytes, but how they help them, that is not really well known or well described. Recently, we have prepared, or we finished another study, which was trying to find out how much plants rely on AMF across the much broader gradient, which included also the forest zone within the Kashmir, Himalayas. So here we have samples from 2,000 to 6,000, 4 kilometers gradient. And we found that AMF taxonomic diversity declines, but phylogenetic diversity increases with elevation, which means that harsh environment may restrict AMF. species diversity, but not phylogenetic diversity. We found fewer taxa, but more distantly related at the higher elevation. So this is a study that was done by Estonians, or in collaboration with Inga Hiesalu, our former postdoc, and other guys from this group in Tartu. The different habitats hold a diverse set of AMF communities. And AMF communities become more specialized with increasing elevation. So in lower elevation, we have more ruderal taxa, but higher elevation, we have more specialized taxa. This is like a preliminary data, just depicting the diversity changes across elevation, and it would deserve more analysis, because for every species and individuals, we also analyze the nutrients in leaves and below ground organs to eventually link the diversity of AMF with plant functioning. What is also, do I have still time? Yeah? Oh yeah, it's not that bad. What is also the reason why plants can, so we said that the plants may have difficulties with the recruitment or dispersal, but once they made it through this risky period, through the juvenile stage, they often stay for long. So they persist on a spot for a long time. So this guy is of this size, and the root is the size of the toothpick, 2 millimeters. But this guy is 22 years old. So we can section it, stain it, and we can see that these plants produce one row of early wood vessels, like this, followed by 5 cells of late wood. How it comes? So 6 cell divisions. Kerner wrote in his book that when the temperature of the growing season is between, let's say, 2 to 4 degrees of Celsius, the one cell division may take 6 days. When the mean growing season temperature is 15 to 20, it may take 5 hours. So if he is true, we have 6 cells. Each is produced within 6 days, or it takes 6 days to produce cells. So it is basically 6 by 6, 36 days. And that is the growing season length in this elevation, meaning the growing season when the soil temperature is unfrozen. And that is very important for these plants. The plants can cope with the freezing temperature above ground. You can see these records for measured by TMS, logger, and we can see the temperature above ground, ground, and below ground. And we can see that during the peak growing season, so this is the transition from July to August, the ground and above ground temperature is regularly going down to -4, -6, but below ground temperature remain above 0. And this is the crucial for the occurrence of plants at high elevation, not the above ground temperatures. Plant can stay unlignified. Everything is formed, almost everything, by living parenchymatic cells, even though there must be some lignification. Like you can see here, those vessels have lignified walls, otherwise they would not be able to conduct the water. But one of the reasons why they are remaining unlignified, because lignification means that plants can quickly transport the water. You know, the plants that are in productive environment, they have big vessels, they can quickly conduct the water. But in this environment, maybe they don't want to be lignified. I told you that maybe the lignification is temperature limited, but maybe they want to avoid it, because if they would have enough vessels and a lot of water in their tissue, they could easily get embolized. So we have now collaboration with a physiologist from Spain, And those plants have maybe a combination of different strategies. They behave like ruderal plants, so they can quickly colonize the new territories. They are colonizing the disturbed sites. But once they are there, they behave like a stress tolerator. So they combine those strategies. Maybe we should rewrite the plant ecology textbook and those the grime strategies, because there are definitely the plants that can behave as a ruderals, also behave as a stress tolerators. And from the physiological point of view, it's very interesting that those plants can have very high photosynthetic rate when the conditions become favorable. And at the same time, they can desiccate, extremely desiccate. They can lose up to 70% of water, and they can resurrect. So maybe this is something that would really deserve more. So age is something that this is really important. It's rather tedious to prepare it. So because you are using the microtome with terribly sharp Japanese disposable blades. So you can see that I'm not doing this work because I have all fingers. So this is done by our very skillful lab technician, Veronica, Eva, Iveta. And once we have the information about age and anatomy of species, we can produce something like atlas, where we can find species with information on anatomy and ecology and a bit of biogeography. Microtome, let's go quickly over. And then we can actually test where we can find the longest living species with the highest longevity. So and we found that the plants with the highest longevity are found in subnival zone, in screes. Those are the cushion plants or the plants with deep taproots. And then we can put it under the test, like we did. We have different predictors for longevity. We use habitat preferences, like species ecological indicator values a la Ellenberg. We use information on size, growth, form, and on top, we use the functional traits. And we found out that altogether, those variables can explain up to 50–55% of interspecific variation in plant longevity. And one of the very important factor is the growth rate. And there is a clear trade-off between longevity and growth rate, something that you know, if you live fast, crazy, it's likely that you would not make it to a long age, and vice versa. And we found this negative relationship between longevity and growth in alpine plants in Himalayas. But if we look at the other species, like we did this cross-analysis for over 4,000 species, we will find the same negative relationship. But interesting is that these residuals from the line can be pretty much predicted by the growing season length, basically by the productivity. We had species from Sahara to Arctic. Usually, if we divided those species into biomes, they have the same regression slope, but they differ in intercept. And these differences are pretty much related to the energy and water availability, which is basically related to the growing season length. The plants also differ in growth. Again, like with the age, the slowest growing are cushion subnival plants and vice versa. And the growth rate seems to be pretty much related to the leaf nutrient economy or root nutrient economy, and also the carbon economy, especially the concentration of soluble sugars. Interesting topic is to look at the biotic interaction, because what I said so far may be heavily influenced species diversity, species distribution by biotic interaction. Just a few examples. Traditionally, what is studied in Alpine is the interaction between big plants and small plants. Those big plants are often represented by the cushion like this. And they are very attractive. They are sort of self-recycling units. they may have higher temperature, they are litter and nutrient trap, and they lure other species that may find a different kind of protection. So we look at that, and the classical approach is that we count what is on the cushion and what is outside, and we compare the situation. Are plots outside and inside differing in species densities, individual plant densities, etc. We did that at 2 different sites, always across the elevation gradient, and we found no support for positive effect of cushion plants on other species. So always there was more species outside than inside. Well, I don't know if I would like to grow in this kind of tough plant. It can be difficult to get through, to penetrate through. So we found little indication for community-level facilitation. So the diversity of species was always higher outside than inside of the cushion. All these stories about the importance or positive effect of big plants on small plants in the alpine zone are very often presented without the context. Yeah. In Himalayas, what we see, the glaciers are retreating and temperatures are rising. Those things are connected, and newly deglaciated substrates is often colonized. And Thylacosphaermum is one of the pioneer. It's one of the best colonizer. And it is often colonizing the best site. And later, it may succumb to graminoids, especially in the lower elevation, in the more productive site, as we can see here on this picture. So this site was deglaciated, colonized by cushion, and is overtaken by graminoids. And this may be fastened by current rising temperature, warming trend, global warming, mind you. But we were also interested if the species that are colonizing the cushion, for whatever reason, are different in terms of traits. If there are some trait syndrome that differentiate them from the other species that do not make it to the cushion. And we indeed found that those plants that are associated with the cushion are unrelated. They are from different lineages, but they share the same traits. They are often rhizomatous, they are fast-growing, they are often dominant in late successional grassland. So it rather seems that this story with the facilitation or not can be pretty much related to a succession. So, and often it's also a size related, especially the older cushion that are senescing, that have some cracks, that have some dead tissue, are more prone to being overgrown by clonal graminoids. But all what I said doesn't necessarily preclude some kind of positive effect that we can see at the individual level. Yeah? Those plants that can finally live with the cushion can get some benefit, even though they are more abundant outside. So we tested that by analyzing the isotopes in stem water and stem tissue or leaf tissue in plants that are growing outside and inside. And the thing is that those big plants have deep roots. And in this dry condition, they can bring the water from the deeper soil layer to the surface. In ecological literature, it is described as a hydraulic lift. And the plants associated with these big guys can profit for that. And we can see it in the differences in isotopic signature, as we found here. But it is context dependent. So we found out that plants associated with the cushion lose in dry condition, so they have better access to the water when they grow without the cushion, and vice versa in cold condition. But is it the Thylacosphaermum really? So this would rather indicate that in some sites, these big guys can provide some positive effect to the other. But in some others, maybe not. Accidentally, we came across the large cushion that were overgrowing other large cushion. So there are two species, Caragana and Thylacosphaermum, coexisting together. They are both considered as a large plant, as a cushion-forming plants, and they were both tested for the positive effect on other species. But here in this context, we found that this benefactor is in fact a strong competitor who is overgrowing the Caragana, as you can see here. And to demonstrate that, we measured the growth rate of the Caragana branches outside and inside of Thylaco. And we found that those that are, of course, covered by, overgrown by Thylaco, they have suppressed growth. It looks like anecdotal, but in fact it is not, because cushion are often presented as a perfect stress tolerator, yes, in the grime strategy. But in fact, they are also super good colonizer of the highly disturbed post-glacial substrate. So they may have a ruderal strategy. So they have seeds that can get easily dispersed. But in some occasion, they can be a strong competitors, like here. So in fact, they combine all three different grime strategies that are described, depending on the context. Yeah. Caragana. Caragana we also tested, we did a case study looking if Caragana has some positive effect. And again, we found that at the community level, there is nothing. The species that are associated with Caragana are more abundant outside. Yes. So at the community level, nothing. But at the individual species level, there was a positive effect, but always at the places where that species was out of the optima, as you can see here. Yeah. And at the individual level, we found a positive effect on the associated grasses in terms of water relationship. The plants that grow inside the cushion, they are both protected against the grazers, and plus they have more water available compared to those plants, the same conspecific individuals growing outside. The same we have demonstrated for the association between the Laemus and Lindelophia. Yeah, you can find that there are grasses inside this perennial Lindelophia. And again, we found that inside we have the same deuterium signature as in water that is 40 centimeters below ground. Yes, and outside there is a signature that corresponds to the upper soil layer, which pretty much indicate that those plants inside can have access, probably through the hydraulic lift, to the water that is in the deeper soil layer. So again, we can use, it's rather tedious to get this information because you have to take a stem, you have to put it to the vials, then cryodistillation in the lab, and isotopic analysis. But we can get closer to the mechanism of how these plants interact by focusing on isotopes like deuterium, like oxygen, or leaf carbon-13. Last but not least, very quickly, climate change and plant responses. This is something that is really a topic in mountains in general, in Himalayas in particular, because the last 3 decades in Himalayas experienced really accelerated warming. What it caused? Few examples. We were lucky by having access to Leo's dataset, and we could revisit the site that he explored. This is an example of Shingola Pass that he surveyed 22 years ago in 2000, and he found only one species because this site was fully glaciated. 15 years after, ice is gone, and we, at the very same site, found 22 species, and under one boulder there was a, how to say, paštika. The wrap of Leoš. So we knew that we are at the site where he worked. Because sometimes those GPS records may not be reliable, but I'm showing particularly this site for this reason, that it is the pass, and we found the garbage he left there, because in these places it decomposes really slowly. So this is the illustration that the plants are really moving upwards, that they colonize the recently deglaciated sites. But this may sound anecdotal. Another approach can be taken using the Landsat images, the remote sensing approach, to look how greening happens or what happens with the vegetation globally during the past 30 years. So Martin Macek, Martin Kopecký played with this data. They calculated NDVI indices over the past 30 years, and just a few most important things that we found out, that not surprisingly, the NDVI, the proxy of productivity, follows the precipitation very tightly. If there is more water in the desert, everything is blooming. And we found a negative relationship with the temperature. So the higher the temperature, less productivity. But this is, again, as always, context dependent. This works very well in the lower elevation desert and steppes, but if we climb to the Alpine, it's actually the reverse. So the more precipitation mean more water, more extreme events, higher soil disturbances, and less cover, not necessarily lower species. So we found this at the regional scale using the Landsat images. And is this reflected at the level of individuals? If we take the annual increments, the growth increments, year-to-year changes in growth, radial growth, can we see something similar if you look at the plants that grow in the drier, mesic, and cold and wetter situation? Is this following this global pattern as indicated by NDVI analysis? We did that by selecting Potentilla pamirica, and we reconstructed the growth and recruitment for the past 60 years. So we took over 1,000 individuals, everything we sectioned from 12 sites, between 5.3 to 5.9 across almost entire elevation range, including steppes, alpine, and subnivel zone. Just briefly, we found out that there is a strong climate control for recruitment and growth, both, but with opposing responses to warming along elevation gradient. So growth is stimulated by warmer summer in colder sites, but reduced by higher precipitation in colder sites, and vice versa for the population in the lower, drier, warmer steppes. Yeah? So something similar that we see at the regional scale using remote sensing technique, we can derive from the tiny plants the size of the roots of Potentilla pamirica is like this, and you can count up to 80 rings. So strong control, good correlation, good match between summer temperature and radial growth in the alpine zone, for precipitation in the steppe zone. I'm not going into details. Another example, just quickly, that beside these approaches, NDVI, radial growth, we can of course look at the community level responses. For that, we established the plots in the alpine and subnivel zone in 2009. And we were lucky that, well, lucky, people were not lucky, because in 2010 there was flash floods in early August that destroyed 1/3 of the capital, the city of Le, and there was almost 300 casualties. People dead, buried during the night. And our plants in our plot suffered. So we found in plots that we have in alpine and subnival zone decrease in vegetation cover, changes in species composition. Species from rather wet habitats increased, while species with optima at higher elevation decreased. So those typical alpine species somehow didn't like it, but species bound to the riverbeds profited. And this was also reflected at the level of traits, that species with adventitious roots profited, by species, small non-clonal plants with main taproots get reduced. So those changes that we see are complex, and it depends on the level that we look at, right? New research avenue, and that's the end. So we are particularly interested in our current project in the multitrophic diversity responses, including plant communities, soil crust, bacterial communities. We are using the moraines as a sites to study the primary succession. Again, this can be pretty much context dependent. The succession is particularly slow in high elevation. Those deglaciated substrates that are close to 6,000, and rather fast at the lower elevation. There is an entire sequence of changes, including the soil crust, cyanobacterial biomass, bacteria. I'm not going into details, just to tell you what we are currently doing. Also, the complexity is increasing. Those pioneer cyanobacteria are very simple, unicellular, while those later successional cyanobacteria are filamentous. In both cases, if you look at the bacterial family richness or plant species richness, there is an increasing trend with increasing age of the substrate. We are doing metagenomic of the root-associated and soil-associated bacteria for different habitats, arid, grassland, subnivel. And there are clear responses. For instance, those grasslands, you can see the unimodal response. So bacteria are increasing and then decreasing towards the mountaintops. We are also interested in age-related trends in biomass and nutrient allocation, because age is something that may not necessarily give us the same outcome as size, because some processes are not really size-dependent, but age-dependent, like reproduction. So it's important when we are studying the plant adaptation to include the age information. Last but not least, we are turning more into physiology. Last year, we were trying to measure the plants with the Lycor at 5.5. Those Lycors are designed to work up to 2,000 meters. So for the first time, so there were days when it went smoothly. There were days where it went not smoothly, and we were really crazy. This work is done together with the Chauma Flexas from Mallorca. And that's it. Thank you for your kind attention.
Many thanks for this presentation. You collected a huge amount of data, sampling, measurements, and we have to digest, I guess, a little bit, because it was full of information.
You know, if there are no ideas, there must be data. Yes, please. That's a good question. Yeah, the Alps are cold, not necessarily dry. That's one important difference between the Ladakh and Alps. Even though we are trying to have like a data from Alps, so last 2 summers, especially during the COVID time, we did a lot of sampling in the Alps. And the Alps, you know, are the site with the millennia-long impact of human, and we cannot say anything the same about the Ladakh high elevation sites, you know. So that's also one of the large difference that in the Alps, everywhere you come, there are people and there are the vegetation is somehow affected. But okay, so this is different. Yeah. Well, there is a lot of things different, of course. Like we have this beautiful gradient that includes also the steppe and desert, so we can directly compare anything with the Alpine. In the Alpine, you have forest underneath, so that's also different. Yeah, of course, it's easy to do research in the Alps. But other thing maybe, which for instance is worth mentioning, especially from this microbial point of view, in the Alps, it's everything heavily impacted by nitrogen pollution, and many of the associations do not exist anymore. So this dependency of plants on microbes and nitrogen is somehow not anymore that strict in the Alps. So at least that's what the microbiologists say. So that's why we were focusing on the plant-microbe interaction in the Alps and in the Himalayas to basically see how is the situation in the pristine environment and in the polluted environment. Yes. We do not have a ski resorts in Ladakh, unlike in the Alps. So this is also somehow reflected in the differences in diaspore load and invasive species, and another thing is we had a side project on the, for instance, the antibiotic contamination in the soil, which is so the bacteria are not anymore resistant to antibiotics in the Alps, so the soil are not pristine anymore. But in Himalayas or in our study regions, still they are responding. So it's and of course this the double stress, drought and cold. This is something very interesting. This basically leads to maybe discovery of some new plant strategies, which you may not find that clearly in the Alps. So I would like to study this, especially this trade-off between the desiccation and photosynthetic rate, or which the plants in cold and dry mountains have to cope with. You know, so this is something that would deserve more research, and I was 2 weeks ago in Kilimanjaro to collect the plants that are basically having the same troubles. They are growing in the cold and dry environment. So maybe we can have something parallel to our data from Himalayas. I don't know. I would have to think. I'm tired to think. Yeah, we did that last year with those Alpine plants that where we measured the photosynthetic rate, this desiccation, the pigments. So we also measured the canopy leaf temperature.
Thank you for
Thank you. Yeah. Thank you for this question, because this is really something important that is going on, and it is a, I would say, rather terrible trend over the past 10 years. And it is pretty much related to the effort of the Indian government to connect each and every village in Ladakh by road. Ladakh was the place where people were coming for hiking without the roads, without the paved roads. So nowadays they are in a haste building the new roads, and the result is that everything around the roads is terribly overgrazed, and places where are far from the road are actually less grazed. So this kind of polarization that we can see in the modern landscape, abandonment on the one side and intensification of agriculture on the other, hit the Ladakh very recently. And you have places that are really terribly overgrazed because there is the road with the asphalt, the paved road. And those traditional pasture lands where young guys had to spend the summer are getting abandoned. Yes. So this is what you see in Ladakh. This is what you see in southern slopes in Nepal. There were lush grasslands that with no yaks, and we were asking the shepherds in the nearby cottage why they are not bringing the yaks to that. They say, no, but there is no service anymore. There is no signal for our cell phones, so we don't want to live there. Because there is no signal. You know, this is what is happening in Ladakh. This kind of globalization and road construction leads to intensification of agriculture along the roads and abandonment on the other. And it is not only the yak, it's rather related, yak too, but also to Pashmina goats. Pashmina goats, there are special goats that have very fine wool at the belly, which is called Pashmina. And this is one of the major industry in Ladakh. It's very expensive, whatever, made of Pashmina. And they have them in high stocking rates, especially around the villages, and everything is grazed like grazed. Yeah, thanks. No, it is not a problem. It is not a problem, and I think this CO2 limitation is not really the issue. There is a Kerner's book, the new edition. There is an entire chapter about the CO2 things and increase and rise and how it affects the plants and experiment, how they simulate it more. No effect. Very few. So this is not really the issue. What is the problem is the photosynthesis is not really that big issue. Like, it's important to study it, for instance, in link with the desiccation tolerance. Yes, but I think Kerner was saying once that we can have a better information if we come with a ruler rather than with a gas analyzer. So if we measure the growth biomass formation, we can get better information about the plant behavior. I don't know. I don't think so. I don't think that, but yeah. It's also a little bit out of my expertise to say. But what I read was that this is not really that limiting, that important. Mm.
When we compare the number of species in Cobrasia in Himalaya with the rest of the world, it's absolutely distant. Similarly, in the case of Saussure. And what is the reason why directly these two genera so many specified in this area?
Well, I don't know. Maybe is it also partly related to the size differences? Like the Himalayas, Senzulato are 15 times bigger than the Alps. So there is more space for like a speciation. Like what I was interested, you often those...
While maybe few in the rest of the world. And I think similarly in the case of Saussure.
That's a good question. Yeah, maybe one of the reasons is that if you have everything at the very limits, water, temperature, what is my experience that if you cross the range, one range, you enter into something that physiognomically looks similar, but you have different species. So there is this kind of fine-tuning that may eventually lead to a new species. So this is, you know, like if we increase 20 millimeters of rain here, nothing will happen. But in this environment, it means a lot. So that's why you have different communities. They are still steppes, but you have the main players of different species. So it's different Artemisia, different Saussure, and different Combrasia in the southern Ladakh than in the northern Ladakh. Of course, there is some overlap, but very often those dominants are different species. But it's still steppe. But you know, so I don't know if this heterogeneity could, and also bedrock. You know, this is also something. These Zanskar, the central Ladakh, made of sedimentary rock, uplifted from the Tethys Sea. That is different from those other mountain ranges. So at the same elevation, you have higher diversity. There is probably higher water available in the soil. The valleys are narrow, those like narrow canyons. They have better microclimate. So and there you can find again Saussure, again Artemisia, but different species than if you cross the range, and you know. So maybe this size and diversity of bedrock and an overall limitation by water scarcity and low temperature leads to something new, the new speciation. And also the stability. Stability is something that plays a crucial role. So maybe this, you know, if something is stable for long, it leads to speciation, like we see on Mount Cameroon. The lowest elevation, which were never dry during the Quaternary fragmentation, have the higher number of endemic species, not the Alpine zone. Here, we have also the high number of basically endemic species or new species on this high elevation plateau, which is dry. The glaciers start at almost 6,000, so they cover only those mountain tops, and everything underneath is basically stable for a long time. So that's probably, or that's how I explain why there are different species or new species or endemic species. So this stability, it's relatively flat. So there is some solifluction and so on, but it's not really that disturbed, like more steeper sides. Because if you, for instance, enter the Karakoram, yeah, there are bigger glaciers, they descend to lower elevation, and there you will find less of those peculiar taxa. And there is, for instance, Saxifraga oppositifolia, like in the Alps or in the Arctic. The only place where you will find it, at 5.5, 1,000 meters higher than Kerner described it in Europe, in the Dome. Yeah. So I don't know. Maybe this dryness also leads to stability, because there is no this advancing and retreating glacier as you have in wetter mountains. Yeah. Indirectly, we were looking at the pigments in the cyanobacteria, in the soil phototrophs. We did screening of the pigments, and we related it to the total solar radiation, not specifically to... this is a good question, but not directly. So, not that we would focus on plants that should evolve, like those white plants, woolly plants with the bracts and so on. I know about a couple of studies from Tibet by Chinese because they are crazy, they are doing everything, but we didn't. But pigments. Last year we were measuring the pigments, yeah. Yeah, like anthoxanthin, flavonoids, and those. Thank you.
Once again, thank you very much.
Thank you too. Thank you.